Hi, I am Priyanka giving a lecture on monoclonal antibody production. So, what are monoclonal antibodies? Before giving any explanation about monoclonal antibodies, first we know about what are antigens and antibodies. Antigens are any foreign particles such as bacteria, virus against which antibodies are produced in our body. Antibodies basically are two types, that is polyclonal antibodies and monoclonal antibodies. Difference between two antibodies is that polyclonal antibodies are not highly specific. Means they are formed or produced against different epitopes of antigens. While monoclonal antibodies are highly specific, means they are produced against a single antigenic determinant or epitope. So, monoclonal antibodies are highly specific as they produced against the antigen or antigenic epitopes, means that is single antigen or antigenic epitopes. They are highly specific, so they are used for various therapeutic or diagnostic purpose. We can understand the basic of difference between monoclonal and polyclonal antibodies such that this is a antigen having three different epitopes, that is 1, 2, 3. When inject into a mouse, if isolate serum from that mouse, serum containing a polyclonal antisera, means it contains antibodies specific against different three types of epitopes or three types of epitopes of antigen, that is 1, 2 and 3. If we isolate spleen cells from that mice, these are spleen cells, and then fuse with myeloma cells, hybrid cells are formed. One, two, three. Then these screening for these hybrid cells takes place, in which only the hybrid cells which produce desired antibody against a particular antigen is cloned, and these clones produce the antibodies that are known as monoclonal antibodies, means these are specific for antigen 1, these are specific for antigen 2nd, and these are specific for antigen 3rd, means these are specific for particular epitopes of that antigen. If we isolate serum, then it contains both three types of antibodies. It is known as polyclonal antisera. But when we inject into mice and isolate spleen cells from that mice and fuse with myeloma cells, then hybrid cells are produced. After a screening process in which only hybrid cells that produce the desired hybrid cells which produce the desired antibodies are separate out. These antibodies are known as monoclonal antibodies or MABs. So, as these antibodies are highly specific and used for many diagnostic and therapeutic purpose, there is need for the production of monoclonal antibodies in vitro. For in vitro production, scientist's name is George Kohler and Milstein derived a method for monoclonal antibodies production in 1975, and this method known as hybridoma technology. So, what is hybridoma technology? In hybridoma technology, first we inject mice with a particular antigen. This is the antigen. Inject mice and isolate spleen cells. This is a culture of spleen cells. Then these are cultured myeloma cells. Suspension of myeloma cells. Then fuse both of the cells. After fusion, 3 types of cells are present in culture, that is unfused spleen cells, these are hybrid cells, and these are unfused myeloma cells. Only the hybrid cells are desired, so transfer these cells on selective media for selection of hybrid cells. In selective media, only hybrid cells are survive and unfused cells die. Then screening process take place in which desired hybrid cells are select. These are desired hybrid cells that produce antibodies of our interest. Then cloning take place. So, these hybrid cells produce antibodies that are known as monoclonal antibodies. So, this is a process by which George Kohler and Milstein produce monoclonal antibodies known as hybridoma technology. In this, first antigen inject into a mice, then isolate spleen cells from the mice. 2 types of cells are used in hybridoma technology. First is spleen cells and then second is myeloma cells. Spleen cells are antibodies forming cells and myeloma cells are cancerous cells. These spleen cells, that is antibody producing cells, die after a certain period of time in vitro culture. So, these are fused with suspension of myeloma cells. Myeloma cells are immortal cells and they have the property of indefinite growth. So, after fusion, three types of cells are mainly produced. First, unfused spleen cells, hybrid cells, unfused myeloma cells. Only hybrid cells are of our interest, so we transfer these cells on a selective media. In selective media, only hybrid cells survive while unfused cells are die. Then after selection of the hybrid cells, a screening process takes place. In screening process, we select only those hybrid cells that produce antibodies of our interest. Then cloning of the desired hybrid cells take place. After cloning, these hybrid cells produce antibodies which are of our interest. So this is the whole process of monoclonal antibody production. After formation of hybrid cells, there is a process known as selection of hybridomers. This is an important process by which we select hybridomers. In this process, spleen cell, these are myeloma cells, fusion, hybrid cells, unfused spleen cells, and unfused myeloma cells. As we discussed earlier, how we select these hybrid cells from the unfused cells. For this, we take selective medium, transfer these cells into selective medium. So what is this selective medium? Selective medium is a medium which contains antibiotic aminopterin. What is the function of these antibiotic? This antibiotic aminopterin only selects the hybrid cells, while the unfused spleen cells and unfused myeloma cells are die in this medium, because this medium contains aminopterin blocks de novo synthesis pathway of nucleotide synthesis. In this method, for selection, we made myeloma cells HGPRT negative. HBGRT is an enzyme, and the spleen cells, these are HGPRT positive. After fusion, the hybrid cells are HGPRT positive. So the hybrid cells survive in this medium, while unfused cells are die in the medium, because spleen cells contain HGPRT enzyme, while myeloma cells contain no HGPRT enzyme. This enzyme is responsible for nucleotide synthesis pathway known as Salvage pathway. Because hybrid cells that contain HGPRT enzyme, they survive in this medium, while the unfused cells die in the medium because they are HGPRT enzyme negative. So in this process, we select the hybrid cells from the unfused cells. This is the process of selection of hybridomers. Now, application of monoclonal antibodies. As monoclonal antibodies are highly specific, they are used for diagnosis of various diseases. They can detect antigens such as detect various type of bacteria, virus or parasite. As monoclonal antibodies detect various antigens, these can detect various types of bacteria, virus or parasite. So these are used for the diagnosis of various types of bacterial, viral or parasitic disease. These are also used for tumor detection. How these antibodies detect tumors? Tumor cells contain some specific antigens which normal cells not contain. So after detecting the antigens present on tumor cells, monoclonal antibodies differentiate between tumor cells and normal cells, and tumor cells are detected with the help of monoclonal antibodies. Another application of monoclonal antibodies, drug targeting. With the help of monoclonal antibodies bound to a specific drug, then the drug can reach to a target cell with the help of monoclonal antibodies. So these are various applications of monoclonal antibodies. Thank you.